All right, so after we have activated our various cell types, our helper T cells, our cytotoxic T cells, and our B cells, what do they actually do, right? What are they actually doing? What's the effector response, right? So what is the end result of them becoming activated, right? And for some of these, we kind of already touched on it, right? But we're going to just review it here briefly, okay? So our helper T cells, right, whenever they become activated, remember, these are CD4. They have the CD4 molecule. They're going to bind with MHC2, right, on our antigen presenting cells. They will become activated, and when they activate, they turn into the activated helper T cell and the memory helper T cell. And really, these have an extremely important role to play with both the cell-mediated immunity and the antibody-mediated immunity, right? So as we talked about, their interleukin-2, right, those cytokines are going to go and activate cytotoxic T cells, right, to become activated cytotoxic T cells, but they're also acting to activate our B cells, right? So with our antibody-mediated immunity, right? So you have to have your helper T cell for the coordination of both parts of the immune system, right? They're also acting on some parts of our innate immune system. If you wipe out the helper T cell, right, and thus you affect you then affect the components of the cell-mediated and antibody-mediated immunity, you've drastically taken a shot to your immune system, right? And so this is exactly what happens in HIV and AIDS, right? And so we'll talk about this here in just a little bit, but HIV is the human immunodeficiency virus, and then AIDS is acquired immunodeficiency syndrome, right? And so you can have HIV without AIDS, but you can't have AIDS without HIV, right? But the way that this works is that it targets and it destroys, the virus destroys our helper T cells, right? Our helper T cells. So let me advance here. For our cytotoxic T cells, right? Remember, after they become activated, they're activated by those helper T cells that we just talked about. They divide into activated cytotoxic T cells and additional memory. I'm sorry, not helper. Activated cytotoxic T cells and the memory cytotoxic T cells. They travel to the infection site, and their end result is they release these things called perforins and granzymes, right? So perforins, I think we talked about this a little bit in our kind of innate immune system. But the perforins, essentially think of this as the word perforate, right? So they're making these openings or these holes inside an infected cell, right? And so again, these cytotoxic, these are CD8, and they're interacting with MHC1, and so these are on infected cells, abnormal cells, tumor cells, things like that, right? So cells that they deem as unhealthy, infected, right? So the perforins put these holes, right, these perforations, if you will, in the cell, and then these granzymes are these cytotoxic chemicals that induce apoptosis, or this programmed cell death on the inside of the cell, and thus destroy the cell itself, right? So through this perforin granzyme activity, right? So you have your helper T cells, your cytotoxic, and then you have your B cell activation, right? So B cells are activated again through the activity of the helper T cell, right? The activated helper T cell with those interleukin-2 cytokines. And again, when the B cell is activated, it begins dividing, that clonal selection, and it begins turning into plasma cells, and the plasma cells produce the antibodies, right? So you can kind of see this schematic here. B cells turn into plasma cells produce antibodies, right? And so these antibodies are freely circulating. Remember, the circulating antibodies have the same receptor specificity as the original B cell receptors, which are also antibodies, and they circulate in the blood. They are at various levels, right? So antibody titers, so the circulating blood concentration of antibodies against a specific antigen that confers some amount of immunity. And again, these circulating antibodies are different than the just basic receptors on other cells in the fact that they're circulating in the blood and they can encounter pathogens as they pass them in the blood circulation, right? And so they're different in that case, in that sense, than the bound receptors on our B cells, right? So speaking of our antibodies are also called immunoglobulins, right? And so when we did our blood lecture, right, we talked about different types of plasma proteins, and one of those were globulins, and one type of globulin is the immunoglobulin, right? And so these can serve to tag pathogens. They can also serve as the receptors, right, as we've discussed. They basically all have the same kind of general structure. They have what are called heavy chains on the inside, lighter, smaller chains on the outside. They have a antigen binding region in a region called the variable region that changes based on the specific antibody that you're talking about. That's specific to various types of antigens, and then there's also a constant region as well, right? So if you look at an image of an antibody, it kind of has this characteristic kind of Y shape, right? So here are those heavy chains on the inside, smaller light chains. This region down here is called your constant region, meaning it stays constant. It doesn't change a lot among different types of antibodies. But then you have your variable region, right? And importantly, the most important thing is in the variable region, you have your antigen binding site, right? So that's where the actual pathogen, the antigen, whatever it is, that's where it's actually going to bind onto the antibody itself, right? So think of that region in this sense, right? The shape of the antigen has to be able to fit, it has to be complementary to the shape of the antigen binding site, right? And if it doesn't bind to it, if it doesn't fit, then it's not going to bind to it, right? And so consider that whenever you are thinking of the specificity of these antibodies, the antigen binding site has to match up with the actual antigen that it's binding, right? So for our antibodies function, they go through these kind of basic processes called neutralization, agglutination, and precipitation, right? So you can see these processes here. So neutralization, the antibodies are going to cover a pathogen, right? They bind to these very small epitopes, right, on the antigen itself, and this essentially kind of neutralizes the pathogen. So whether it's a virus or a toxin or a microbe or bacteria, whatever it happens to be, it essentially neutralizes, hence the name, neutralizes that pathogen, right? The next one is agglutination. Think of this as like clumping together. So multiple antibodies can bind to several different pathogens in different regions, thus causing clumping. This is really helpful for bacteria, right, or acting on bacteria. And then you have precipitation, right? And so precipitation are these cross-links among antibodies and antigens that form these large complexes. And then these complexes settle out of solution, so they're insoluble, and then they can be phagocytosed by some type of phagocyte, so a macrophage or neutrophil or something along those lines, right? And so these are, again, this is specifically for these circulating antibodies, right? Not the ones that are bound to the B cell, but these that are circulating, that have been produced by our plasma cells, right? The constant region of the antibody can also be involved in these various processes here, right? So you can read about them. We can look at the similar types of images here. So one called the complement fixation. And so we talked about this whenever we did our innate immune system. We talked about the complement system, those various proteins circulating in the blood that are ultimately going to produce that membrane attack complex, right? But some of the complement proteins can bind to that constant region of the antibody, right? They can also perform something called opsonization, where they bind to receptors of phagocytic cells, triggering phagocytosis, so that engulfing of cells, and then they can also stimulate the release of cytotoxic chemicals from cells like natural killer cells, right? So that same kind of perforin and granzyme process that we talked about when our cytotoxic T cells become activated, right? Here you can see various types of immunoglobulins, antibodies. So I'd like you to be familiar with them in the context of the various types. So you'll all see them abbreviated IG. That just means immunoglobulin. So immunoglobulin A, antibody A, D, E, G, and M, right? And these you find in different areas performing different functions, right? So IgA you find in lots of secretions, right? So saliva, tears, breast milk. This is actually the one that confers immunity to newborns through the breast milk, right? So some passive immunity there. IgD, this is the B cell receptor, right? So that characteristic Y shape that we talked about earlier. IgE, this is involved with allergies, asthma, and parasitic infections. IgG, secreted by plasma cells in the blood, and this can also cross the placenta, again conferring some passive immunity to the fetus before birth. And then IgM may be attached to the surface of B cells, secreted in the blood, things like that, right? And so lots of different types of antibodies, each having kind of their own respective function. Okay, so let's stop here. You can consider some questions. We'll pick up in the next part, beginning to talk about memory, and then we'll talk a little bit about kind of sensitivity reactions. We'll talk a little bit about vaccines, and then we will be done.